Hi. Once again, let's add carbon to water, to H2O. But this time, we'll add a second oxygen as well, which forms a double bond. The new molecule is HCOOH, which is the smallest organic acid, called formic acid. The H is bound to the COOH group, called carboxyl. Acids are generally hydrophilic due to the oxygen atoms. If, however, we replace the H for a CH3, a methyl group, formic acid becomes just a bit more hydrophobic. Methyl groups are hydrophobic. This new acid is called acetic acid, and when it's diluted in water, we call it vinegar and often use it in our salads. Extending the molecule with more and more hydrophobic CH3 groups generates ever-increasing hydrophobic tails linked to the carboxyl COOH acidic group. These molecules are called fatty acids, and when two fatty acids are linked together, we get lipids. Lipids are interesting in that they have long fatty acid hydrophobic tails and a hydrophilic head. The hydrophilic head can be further modified to contain a negatively charged phosphate atom, thus generating phospholipids. Phospholipids are amphipathic in that they have a hydrophilic phosphate-containing head and very hydrophobic tails. When phospholipids interact with water, they arrange themselves so that their polar heads contact the water molecules, and their hydrophobic tails shun away from the water. Think about it. The best geometry to satisfy both characteristics is to form a bilayer of lipids. The heads contact the water, while the tails mingle amongst themselves. Such lipid bilayers are the fundamental structure of biological membranes that surround cells. Inside the cell is where life exists, where sugars, proteins, and even our genes are made and function. The membranes can further divide the cell into subcellular compartments, which we call organelles. The central organelle that contains our genetic material, the DNA, and our chromosomes is called the nucleus. As I mentioned in previous units, prokaryotes, like bacteria, don't have a nucleus. They do, however, have DNA, but it's not packaged in a membrane-bound organelle. As multicellular organisms began to develop and animals and plants evolved, the cells began to take on specialized functions, creating organs such as muscles, livers, lungs, and limbs, or leaves, flowers, branches, and roots. The complexity of life required cells to become more complicated and specialized to carry out different tasks, and for this, special subcellular compartments, organelles, enabled cells to function more efficiently. Each organelle has its own function, and together they create a highly sophisticated molecular machine able to carry out all the tasks of life. The nucleus, as mentioned earlier, contains our genes. Genes are, in essence, blueprints, the instructions for making the proteins of our bodies. However, the synthesis of proteins go on outside of the nucleus on special structures called ribosomes. Often, ribosomes are associated with a network of membranes called the endoplasmic reticulum, the ER. Proteins produced in the ER are then passed on to the Golgi apparatus, the loading dock of the cell. Here, the proteins are shipped off to other organelles, such as lysosomes, or can even be exported out of the cell. All this activity requires energy, and the energy plants of the cells are the mitochondria. All the organelles are bathed in a fluid of the cell called cytoplasm. Now, for a moment of truth. Honestly, when we examine cells under the microscope, we don't really see these structures, the organelles, so clearly, unless we treat the cells with special stains that color organelles specifically. Let's go to the mini class again and see how staining works.